Scientists agree that eradication of HIV requires an effective vaccine. Because HIV mutates rapidly, it has defeated traditional vaccination strategies. However, research at the University of Texas has shown the path to the first effective HIV vaccine. Traditional vaccines are composed of weakened or killed microbes. The vaccine binds and activates human B lymphocytes. This causes cell multiplication, followed by a production of protective antibodies. The process also generates memory B cells that remember the vaccine structure. Upon contact of the body with the infectious microbe, the all-important memory cells respond rapidly and produce antibodies. If sufficient antibodies of the correct type are produced, they coat the microbe, and scavenger cells ingest the microbe, eliminating the threat of infection. No test vaccine based on traditional scientific principles has induced sufficient protective immunity to HIV. Dr. Sudhir Paul's research team has located a tiny, mostly unchangeable region of the coat protein GP120 as the Achilles heel of HIV, shown here as Module A. Without this module, HIV cannot attach to human T lymphocytes and initiate infection. HIV hides this region from the immune system by a clever evolutionary trick. The vulnerable HIV module silences human B cells instead of stimulating protective antibodies, thus fooling the immune system into becoming tolerant to HIV. Except for rare survivors of prolonged HIV infection, humans do not produce sufficient antibodies to the vulnerable module. Conventional test vaccines are also ineffective in stimulating the antibodies. As the immunity is inadequate, human T cells exposed to HIV are infected. These cells are essential for immune defense against opportunistic microbes. Therefore, HIV causes acquired immunodeficiency syndrome, also known as AIDS. Dr. Paul's research team has developed a prototype vaccine that stimulates protective antibodies to HIV. The vulnerable module A of GP120 is activated chemically to become exceptionally sticky and bind covalently to B cells, a process involving intimate sharing of electrons. Instead of inducing tolerance, the binding energy released from the chemical reaction with cells is used to stimulate production of antibodies to the vulnerable HIV region. A second stimulatory module also helps overcome the tolerance. As a result, memory B cells are generated that can produce protective antibodies when exposed to HIV. The prototype vaccine is designed to produce antibodies with enzymatic activity, or ABZymes. A single ABZyme is reused repeatedly to destroy thousands of GP120 molecules. Compared to conventional antibodies that bind HIV on a one-to-one basis, ABZymes can produce powerful protection against HIV. In animal tests, the prototype vaccine has produced antibodies that block infection of human cells by diverse HIV strains from around the world, a long-standing goal in HIV vaccine development. The next steps are maximizing the vaccine-induced immune response and human tests for effectiveness and safety. Visit covalentimmunology.org to learn more.